Dear students, in last class we are discussed about some techniques of electrophoresis, that is free flow electrophoresis, micro electrophoresis, moving boundary electrophoresis and zone electrophoresis. In zone electrophoresis we discussed only paper electrophoresis. Today let us discuss about cellulose acetate electrophoresis. So, it is a modified version of paper electrophoresis developed by Kahn in 1958. In this type of electrophoresis, bacteriological acetate membrane filters are taken in place of regular chromatography paper. In cellulose acetate electrophoresis, we have to use a bacteriological acetate membrane filters for the suppression of component mixtures, and the procedure of cellulose acetate electrophoresis is same like that of paper electrophoresis. The advantages of cellulose acetate stripes over chromatography paper is the cellulose acetate stripes are chemically pure and free of lignin and hemicelluloses and generally acts as barrier in free movement of large molecules, and because of low content of glucose, cellulose acetate stripes are suitable for electrophoresis of polysaccharides. Cellulose acetate is not hydrophilic, and this holds very little buffer, which further helps for a better resolution in a short time, and it contains 2 to 3 acetyl groups per glucose unit, and its absorption capacity is less than that of paper, and it gives sharper bands, and it provides a good background for staining glycoproteins. Next, applications of cellulose acetate electrophoresis. So, it is especially used for clinical investigation such as the suppression of glycoproteins, lipoproteins and hemoglobin from blood. Next, advantages of cellulose acetate electrophoresis. Here no tailing of proteins or hydrophilic materials, and it is available in wide range of particle size and layer thickness, and it gives sharp bands and offer good resolution, and here a high voltage can be applied which will enhance the resolution. And next, disadvantages of cellulose acetate electrophoresis. So, it is expensive, and in presence of sulfonic and carboxylic residues causes induced electroosmosis during electrophoresis. In this picture we can observe a apparatus of cellulose acetate electrophoresis. So, this is the schematic representation of cellulose acetate electrophoresis. In this picture we can observe the setup of cellulose acetate electrophoresis. So, here the setup is same to that of paper electrophoresis, except the acetate membrane filter paper. So, in cellulose acetate electrophoresis, so we can use bacteriological acetate membrane filters other than chromatography papers. Next, gel electrophoresis. Gel electrophoresis involves the use of gel as supporting media for suppression of DNA, RNA or proteins under the influence of electric charge, and it is usually performed for analytical purposes but may be used as a preparative technique to partially purify molecules that prior to use for the other methods such as mass spectrometry, PCR, cloning, DNA sequencing and immunoblotting. This is the most commonly used electrophoresis in biotechnology laboratories and is used for almost all types of experiments. In this picture, we can observe the apparatus of setup of apparatus of gel electrophoresis. Next, let us move on to the principle of gel electrophoresis. When a potential difference is applied across the electrodes of horizontal electrophoretic tank containing agarose gel and biomolecules such as nucleic acids are loaded, and then they get separated according to their molecular size. Here the bigger molecules have more molecular size and smaller molecules have smaller molecular size and move to their respective electrodes. Here the agarose gel acts as a sieve. As in a sieve, the large particles stay above and the particles which are smaller than the pore size passes through it. Similarly, in the gel, the larger and the bulky molecules stay behind, whereas the smaller molecules move faster and quickly towards their respective electrodes. This process may be imagined like a running competition. The one who is thinner and have a flexible body will be at the ending points of ending points. They move faster than the one who is fat and bulky. In this picture, we can observe the apparatus of gel electrophoresis. Here we can observe a power supply, a cathode and anode electrodes, and agarose gel and electrophoretic buffers and sample well. And here in second picture, we can observe the larger particles stay above, and the particles which are smaller than the pore size passes through the agarose gel, and in the gel the larger and bulky molecules stay behind, or they stay above, and whereas the smaller molecules move faster and they move quickly towards their respective electrodes, and the smaller molecules are run fastly than the fat and bulky molecules. Gel electrophoresis that involves the use of a gelatinous material such as agarose, acrylamide, starch or cellulose acetate as the matrix. The gel acts as a support medium for the sample, and gels are used to separate samples containing proteins or DNA. In that, first one, starch gel. In starch gel electrophoresis, a swollen potato starch granules are used as a supporting medium, and a suspension of granular starch should be boiled in a buffer to give a clear colloidal suspension. The suspension on cooling sets as a semi-solid gel due to intertwining of the branched chains of amylopectin. In order to avoid swelling and shrinking of these potato starch granules, petroleum jelly is used, and starch matrix is suitable for isoenzymes. So this starch matrix is used for the separation of isoenzymes, and the gels are more opaque than acrylamide or agarose gels. By using starch electrophoresis, non-denatured proteins can be separated according to their charge and size. They are visualized by using naphthol black or amino black staining. Next, advantages of starch gel electrophoresis. It has a highly resolving power, and sharp zones are clearly obtained. The components resolved can be recovered in reasonable yield, especially proteins, and it can be used for analytical as well as preparative electrophoresis. Next, disadvantages of starch gel electrophoresis. It is highly it shows highly electroosmotic effect, and it has a variation in pore size from batch to batch. Next, agarose gel electrophoresis, or here in agarose gel electrophoresis we have to use agarose gel as a solid support medium. So agarose gel is a natural linear polymer extracted from seaweed that forms a gel matrix by hydrogen bonding when heated in a buffer and allowed to cool. Agar is a mixture of polysaccharides extracted from seaweeds. Agarose is a highly purified uncharged polysaccharide derived from agar. Agarose is chemically basic disaccharide repeating units of 3,6 anhydro L-galactose, and agarose that dissolves when added to boiling liquid. It remains in a liquid state until the temperature is lowered to about 40 degrees Celsius, at which point it gels. The pore size may be predetermined by adjusting the concentration of agarose in the gel. The agarose gels are fragile. They are actually hydrocolloids, and they are held together by the formation of weak hydrogen and hydrophobic bonds. And the pores of an agarose gels are large. Agarose is used to separate macromolecules such as nucleic acids, larger proteins, and protein complexes. Agarose gel electrophoresis is a powerful suppression method frequently used to analyze DNA fragments generated by restriction enzymes. In this picture, you can observe gel structure of agarose, and the separation medium is in a gel made from agarose, which is a polysaccharide derivative of agar, and the agarose gel consists of microscopic pores that act as a molecular sieve which separates molecules based upon charge, size, and shape. These characteristics together with buffer conditions, gel concentrations, and voltage affect the mobility of molecules in gels. So, this is the schematic representation of agarose gel electrophoresis. Here we can observe buffer solution or buffer tank, and an electrode, anode and cathode electrodes, and a sample wells and power supply. Let us know about the applications of agarose gel electrophoresis. Suppression of it is useful for the suppression of restriction enzyme digested DNA, including genomic DNA prior to southern blot transfer. It is often used for separating RNA prior to northern transfer, and it is useful for the analysis of PCR products after polymerase chain reaction to assess for target DNA amplification, and it allowing estimation of the size of DNA molecules using a DNA marker or ladder which contains DNA fragments of various known sizes, and it allows the rough estimation of DNA quantity and quality. And by using agarose gel electrophoresis, quantity is assessed using lambda DNA ladder which contains specific amounts of DNA in different bands, and it is useful for the it is used to estimate the quality of DNA is assessed by observing the absence of streaking or fragments or contaminating DNA bands. And this technique is also used for the DNA separation in DNA fingerprinting. Next, advantages of agarose gel electrophoresis. The advantages are that the gel is easily poured and does not denature the samples, and samples can also be recovered. It is easy to prepare and a small concentration of agar is required for this technique, and the resolution is superior to that of filter paper, and large quantities of proteins can be separated and recovered, and adsorption of negatively charged protein molecule is negligible, and it absorbs proteins relatively less when compared to other medium. Sharp zones are obtained due to less adsorption, and recovery of protein is good, and good method for preparative purpose. Next, disadvantages of agarose gel electrophoresis. The disadvantages are that the gels can melt during electrophoresis, the buffer can become exhausted, and different forms of genetic material may run in unpredictable forms, and here the electroosmosis is high, and the resolution is less compared to polyacrylamide gels, and different sources and batches of agar tend to give different results, and purification is often necessary. And next one is polyacrylamide gels or PAGE. Polyacrylamide gel is made chemically by acrylamide. It is a monomer, and bisacrylamide, it is a cross linker. So polyacrylamide is made chemically by acrylamide and bisacrylamide, catalyzed by an initiator and accelerator. Here initiator is ammonium persulfate or riboflavin, and accelerator is TEMID. The abbreviation of TEMID is tetramethyl ethylene diamine. In this gel, pore size and resolving power is totally depends upon the concentration of acrylamide and bisacrylamide. The concentration of the gel normally varies from 5% to 25%. This gel is used in electrophoresis for the separation of proteins ranging from molecular weight lesser than 5000 to greater than 5000 to lesser than 2 lakh, and polynucleotides ranges from greater than 5 to nearly 3000 base pairs in size. Polyacrylamide has smaller pore size. It is better, it gives better resolution and separates smaller molecules. Here the pore size is about 2 base pair differences in DNA, and acrylamide is synthesized. It is not naturally occur. It is a synthesized form. It occur in we have to prepare this acrylamide. In this picture we can observe this schematic representation of a page that is polyacrylamide electrophoresis. A typical setup consists of a gel slab that is a sandwich between two glass plates with the ends enclosed in upper and lower reservoirs of buffer, and samples to be run are loaded in wells at the top of the gel. In conjunction with the tracking die, an electrical voltage is applied between the upper and lower reservoirs, causing the samples to migrate down through the gel. Once the gel has been run, it is treated to reveal the positions of the samples. Let us know about the components of phage gel. The first one, acrylamide. Acrylamide forms long polymer chain, and next one is APS, that is ammonium persulfate. Here the polymerization induced by APS, which generates free radicals, that is charged oxygens. Next, TEMID. Here TEMID means methyl ethylene diamine. Here TEMD is a free radical stabilizer, and next one is bis-acrylamide. Bis-acrylamide is a cross-linking agent and links along polymers of acrylamide, and here pore size is determined by percentage of a acrylamide and the amount of cross-linker. And the copolymerization of acrylamide with methylene bis-acrylamide produces a mesh-like network in the 3 dimensions consisting of acrylamide chains with interconnections formed from the methylene bis-acrylamide. In polyacrylamide gel electrophoresis, the gel is made from the combination of acrylamide with methylene bis-acrylamide that produces a mesh-like network in 3 dimensions, and these mesh-like network consisting of a acrylamide chains that are interconnected with the methylene bis-acrylamides. Dear students, this is about this today's session. The remaining aspects of phage is we will discuss in next class. Thank you.